Chromatin is the highly organized complex of DNA and proteins that packages our genetic material inside the nucleus. Without this organization, DNA would become tangled, damaged, and difficult for the cell to use. But chromatin is much more than a storage system. It is a dynamic structure that determines which parts of our DNA can be accessed and which remain hidden. To understand chromatin, let's begin with DNA itself. The DNA double helix does not remain as a long, loose thread inside the nucleus. Instead, it wraps around specialized proteins called histones, forming repeating units known as nucleosomes. These nucleosomes are connected by short stretches of linker DNA, creating a structure that resembles beads on a string. However, DNA packaging doesn't stop there. The nucleosome chain folds further into thicker chromatin fibers. These fibers then form loops that attach to a protein scaffold. The loops fold again and again, producing increasingly compact structures until they finally form the familiar chromosomes that become visible during cell division. This remarkable hierarchy allows an extraordinary amount of DNA to fit inside the nucleus while still remaining organized. But compacting DNA is only one of chromatin's jobs. Chromatin also controls gene expression. Not every gene should be active at the same time. Different cells require different sets of genes. For example, a nerve cell and a muscle cell contain exactly the same DNA, yet they perform completely different functions. Because they have different set of genes open for transcription. Chromatin helps determine which genes are available to be read. When chromatin is tightly packed, the DNA is inaccessible to the proteins responsible for gene expression. This condensed form is called heterochromatin, and genes located within these regions are generally inactive. In contrast, when chromatin becomes loosely packed, the DNA is exposed and accessible. This relaxed form is called euchromatin, where genes can be actively transcribed. Cells constantly switch between these two states through a process known as chromatin remodeling. By changing how tightly DNA is packaged, cells can turn specific genes on or off in response to developmental signals and environmental changes. Chromatin remodeling is also essential during DNA replication and cell division. Before a cell divides, its DNA must be accurately copied. After replication, the chromatin condenses into highly compact chromosomes, allowing the duplicated DNA to be evenly distributed between the two daughter cells. Beyond controlling gene activity, chromatin provides another important benefit. It helps prevent DNA from becoming tangled and protects it from physical damage, ensuring the genome remains stable throughout the life of the cell. When chromatin structure becomes disrupted, serious diseases can develop. Mutations in proteins that regulate chromatin organization have been linked to numerous disorders, including many forms of cancer.